Okay, good morning, everyone. Can you hear me okay? Okay, I think we're going to get started. So my name is Jeffrey LeDue. I work at the Center for Brain Health here at UBC in Vancouver. And I'd like to welcome everyone to our UBC Tissue Clearing and Expansion 2021 workshop. Our event today is being hosted by our Dynamic Brain Circuits Research Cluster and another group on campus called the BC Regenerative Medicine Initiative. So before we move into our event, I wanted to stop and acknowledge that the University of British Columbia is located on the traditional, ancestral, and unceded territory of the Musqueam people. If you're curious about this land acknowledgement or why we're doing it, Nicole is going to put some links in the chat that link to UBC resources about this. And if you're curious, it's a good time to take a look, as next week is our first National Day for Truth and Reconciliation. So I just wanted to say a little bit about our context here at UBC before we move on to our workshop. So the Dynamic Brain Circuits Cluster that I mentioned and the BC Regenerative Medicine Initiative, these groups are supported by the UBC Office of the Vice President Research and Innovation through their Grants for Catalyzing Research Clusters program. The Dynamic Brain Circuits Cluster is based at the Center for Brain Health, and BC RegMed is based in the New School for Biomedical Engineering. It's part of our mandate to foster the adoption of new techniques which transform the way we address research questions in neuroscience, but also biomedical research broadly. So both of these groups have had success in securing federal and provincial infrastructure funding, which we're using to support the acquisition of a conventional and lattice light sheet microscopes. Tissue clearing and expansion microscopy have been areas of focus for us for several years. We ran the first workshop as a satellite meeting for NeuroFutures 2017, and we did a follow-up meeting, UBC Tissue Clearing Expansion 2019. Both of these were in person and had hands-on components, but given the situation we're all in, we're pleased that we can continue our series of meetings this year with an online format. So these are all the individuals that are coming together to help us put this on. We're very grateful to everyone on this slide for their participation. These people are donating their time, their energy, and their expertise to make this event happen. And we couldn't do it without them, and we thank them for their participation. So the program that we've put together today will include talks from tissue clearing experts. We will have 2 talks on matters related to tissue clearing and 2 talks related to expansion microscopy techniques, and then a general discussion and wrap up at the end of the session. Tomorrow, if you join us at the same time, we're going to have 3 sessions, and this is where we're hoping to really get into the practical aspects of doing tissue clearing and expansion microscopy. Our first session tomorrow, we will be joined by Brian from Life Canvas Technologies, who will lead a protocols discussion. We'll have a session on imaging of cleared and expanded tissues with Translucence Biosystems and Zeiss, and we will focus on the use of conventional and lattice light sheet microscopes to image these tissues. And then our final session will be on data analysis, and we will be joined by Molly and Maria from Arrivas, who will demonstrate a pipeline that we've been working on to analyze some data with Vision 4D. I wanted to point out that we made an open science framework project to host the materials associated with the workshop. So Nicole can put the link for this in the chat and you can take a look. We've tried to include the relevant papers or links to papers, preprints, protocols, the data and the pipeline for tomorrow, and information about the imaging systems provided by the vendors, as well as the slides for anyone who's willing to share those. So just the usual Zoom housekeeping before we move on. Please keep your mic muted unless you're actively asking a question. If you have a question, you can use the raise hand function, or you can type it in the chat. We'll monitor the chat as we go throughout the workshop. And before we go into the session, I just wanted to thank the people that have been helping me put this workshop together. This includes Nicole, who is a computer science co-op student working with our brain circuits cluster, Lisa, the program manager from BC Regenerative Medicine Initiative, graduate students at our center, Mathias and Sarah, as well as Mehwish, who is a postdoctoral fellow in the lab of Cheryl Wellington at the Center for Brain Health. And at this point, I'm going to turn it over to Mehwish, and she will introduce the topic of tissue clearing and then proceed on to introduce our guest speakers. So Mehwish, do you want to share your screen?
Thank you, Jeff. Indeed, I'll share my screen now. Can you see it clearly? All right, perfect. Good morning, everyone. Thank you so much for participating in this workshop today. And it's an absolute honor to host such giants of the field and to learn from you today, and also to create an opportunity for our researchers at UBC to take this forward. As Jeff said, I am Mehwish, and I work in Professor Cheryl Wellington's lab, and our prime focus is traumatic brain injury. And we've just started looking into tissue clearing, and the possibilities seem endless. So I'm just going to give a quick overview of the topic tissue clearing itself. I will not say a lot, especially in the presence of the experts that we have today. So early 20th century saw the dawn of a new method where tissues and stuff can be made transparent. While this was a very crude and not so sophisticated method initially, but it resulted up opening a new door for viewing the biological specimens and actually looking through them and being able to see things intact the way we haven't done previously. But it wasn't until the 21st century, or let's say early 2000s, until when we were not able to have sophisticated imaging technologies, which actually enable us to go forward and to use this technology. The core principle of tissue clearing is to basically deal with light dispersion, which is because of the biological components of the specimens that lead to a blockage of light penetration through the tissue. So we are unable to go beyond a certain limit. There are various methods that were developed along the way. The initial ones were mainly based on organic solvents. These organic solvents were harsh, they were toxic sometimes, but they do the job. The core steps include, getting the tissue from perfused specimens, and then dehydrating and getting rid of the lipids so that the light can actually pass through them. There's a final step before you can image, which is about matching the refractive index so that you can get your specimen in the best focus possible and can identify your target molecules, either it is proteins or now in many cases even RNA and beyond. Here you can see a few examples of the methods or the adaptations of the organic solvent-based tissue clearing paradigm. Other than that, there were some other methods that were developed a bit later than the organic ones, which were based on aqueous solutions. And these actually provide higher compatibility with immunofluorescence labeling, and also were much more toxic in the choice of reagents that you have to use for the process. Lastly, but not the least, was a modified method of the aqueous-based solution, which is much more friendly in terms of keeping the tissue intact and being able to manipulate it multiple times, which was the hydrogel embedding technique. In this method, there's an additional step where after the tissue is fixed, you put it into a solution that can give it a sort of a pseudo matrix that holds the tissue together intact. And then this is followed by clearing, that is the delipidation process, either through an active electrophoretic intervention or through a passive method. The following steps are pretty much same. You have to align the RI of the sample, and then you can image it with different microscopes. So all these methods in total provided a huge opportunity to not only clear tissues that were soft, for example, the lungs, the embryo, heart tissue, as well as the white adipose tissue, but also tough structures such as bone and teeth. There has been a lot of applications that are now compatible in studying organoid research as well as studying tumorous tissue. So the possibilities are really endless here. Also, the thing that excites me the most about using this technique is that you can now, with all the other methodologies that have been developed over the years, you can actually combine the output of tissue clearing and expansion also with many other methods such as single-cell RNA sequencing and also imaging using magnetic resonance imaging. And the holistic overview that you get of a problem is amazing. One of these methods can actually direct towards the others and vice versa. Here at Wellington Lab, we are focused at looking at the idea of the project that I'm working with is to kind of create a temporal and spatial pattern of neuronal activation after traumatic brain injury. And then we're also trying to understand the context of Alzheimer's disease related components in those mice, and then to build up a link between these and the transcriptomic changes. I will show us a little bit of our data that we have been able to acquire so far. The method of our choice has been the shield fixed hydrogel-based system. And it's a tremendous system in terms of the strength that it gives to the tissue, and the ability that we can do multiple rounds of fluorescence labeling is amazing. It's also very good in preserving the endogenous signal. This tissue is prepared from transgenic mice that express CFOS upon stimulation. And as you can see, the neatness of being able to retrieve the signal after 6 weeks of tissue processing is awesome. We can get a very neat resolution with our light sheet microscope, the Z1, the old one. We haven't not yet tried the light sheet, and I'm excited what that can bring to the table. But even with the old light sheet microscope, we were able to get a really nice resolution with this method. Here's another 3D rendering. On the left side, you see a spinal cord rendering. That's also one of our interests when we're looking at the spinal cord injury. And on the right side is a 3D rendering of a CFOS expression and in a mouse brain. And this was imaged using a modified chamber, which you will hear a bit about tomorrow, how you can image the whole brain in one go. Also, with our graduate student, Honor Sheng, we are optimizing to look at the vasculature of the mouse brain by using retrograde tracers. And here you can see a good depiction of what it looks like in our pilot data. We are able to co-label using 2 different tracers and then to image it in 3 dimension. And to now we're in the phase of actually being able to analyze these steps. And that's how we are also looking forward to learning today from many of you and what are the approaches in your lab. The tool of choice so far for the analysis of these tissue has been ERVIS, and as you will hear more tomorrow, but this is a really fantastic tool in enabling people like me who are not very computationally expert to actually use an interface where you can upload your data and analyze it in 3-dimensional and get quantitative information from a volumetric set. So that's how it is helping us with the project. It is able to give us multidimensional comparative renderings. There's a possibility to register with an atlas to go specific regions and find the information, as well as also to quantify based on hotspots within the brain upon a stimulus.
Good. All right. So today what I'm going to talk about is a tool that we've developed called FastMap. Which is an acronym for a flexible atlas segmentation tool for multi-area processing of biological images. In this field, there's always tons of acronyms, so I guess we just wanted to play as well and contribute one. The work that I'm going to present today is primarily done by 2 students in my lab, Dylan and Daniela. So I just want to acknowledge them up front. So there was a lovely introduction into the field of tissue clearing from Mehwish, and then of course a wonderful presentation from KC right there. So I'm not going to go into this in too much detail, but I think it's important to sort of think about what the field of tissue clearing has really done for the field of neuroscience itself. I think that it's really given us a new view of the brain, and it's allowed us to ask questions more readily about connectivity and interactions between different brain regions. And of course, some of these questions could have been answered before with different methods, but this certainly has facilitated a lot more people asking types of questions. So we can start to look at activity throughout the brain instead of just looking at individuals' regions or going region by region. So this is an example of transgenic CFOS reporter mouse here, so we can see labeling throughout the entire brain. And we can look at other things as well, such as structural connectivity throughout the brain. So looking at the morphology of intact neurons, which is difficult to do in section tissue. We can look at the continuity of vascular systems throughout the brain. Again, this is difficult to do without the tissue intact. And as was mentioned, we can even go down now and look at in situ hybridization to get activity-dependent markers. This is ARC in a cleared brain. And so we can get all of this really strong, diverse information about structure and function of the brain. But we really need tools to handle this. And so how do we make sense of all of this data that we now have? When we started doing tissue clearing, this is really what it felt like to us. We managed to clear some brains, back in 2012, 2013, and we thought, well, that's really great. Well, now what? Well, then we sort of worked on imaging techniques using things like light sheet microscopy, and we managed to get images of the brain, and well, that's great. But now what? And so really we're at the point now where we need tools to analyze these large data sets that we're getting. My lab is particularly interested in looking at brain-wide activity mapping. So we'll use techniques like brain-wide CFOS labeling or something like that. And we'll map the brain then onto an atlas so that we can correlate the activity in different regions of the brain. So we can essentially create a giant correlation matrix here that has the activity in each of, say, 100 different brain regions correlated with the activity in all of those different regions. And by doing that, we can essentially create a functional connectivity network to understand how different areas of the brain are interacting with each other during different behaviors, or in our case, what we're usually looking at is during retrieval or formation of a particular memory. So in order to get to this, really the first step is to have a good, accurate registration of the brain to an atlas so we know where these different neurons are that are active. Now, there's a number of different approaches to do that. This is certainly, what I'm going to show you today is certainly not the first atlas registration tool. There's many of these available. So for example, here, this is something that is based on the Mouse Imaging Center's fMRI or MRI atlas. There are things such as ClearMap from Mark Tessier-Levine's group, and another common one here is the whole brain approach by Daniel Firth. So there's lots of tools that you can actually use in order to get your tissue matched up to an atlas. But some of these, the existing approaches can be computationally demanding. Some require, really large, expensive workstations in order to have the computing power to perform this sort of analysis. Some might require knowledge of a particular programming language that you might not be familiar with. And so they're not as simple as some people might want them to be, including ourselves. And I don't want to give the impression that these tools shouldn't be used. In fact, we do have a different atlas registration pipeline that's modified from this whole brain approach, and we use that when we're interested in looking at large number of regions. But there are a number of scenarios where we felt a simpler tool could be beneficial, and this is really what led Dylan to start developing FastMap. So what is FastMap? Well, simply put, it's an open source atlas segmentation tool. And when we developed this, we really had the ideas of simplicity and accessibility as our central priorities. And so these aspects of the package arise from the fact that the package is constructed as an ImageJ or a Fiji plugin, so it works quite easily across different operating systems. It's simple to install and to use. We also wanted the approach to be flexible so that one tool could be used for a variety of different tissue types or purposes. So you can use different types of biological samples. And there's also flexibility across imaging and tissue preparation methods. This, of course, in the context of a tissue clearing workshop, probably most people might be interested in using this for cleared tissue, but you can also use it with section tissue. You can use it with fluorescent imaging. You can use it with bright field imaging. Anything that you've got, you can definitely use it with that. Now, some of the flexibility here arises from the fact that essentially there is not one fixed atlas that is to be used with this tool. End users are able to create their own atlas plates to upload to the program so that you can use any self-created atlas that you want. And this is really helpful in situations where there isn't an atlas available for the type of work that you're doing, or there simply needs to be changes made to that atlas. So for example, often with cleared tissue, immunohistochemistry or in situ hybridization can be really challenging, and it can also be really expensive. And it's often desirable to work with as little tissue as possible. So for example, sometimes we're interested in just examining different hippocampal subregions. And to do that, we'll dissect out the hippocampus before clearing and imaging, but we still want to be able to register that tissue chunk to an atlas. And that's something that we can easily do with FastMap just by creating a hippocampus specific atlas to upload. All right, to be completely transparent here, there's a few things that FastMap is not. It's not a fully automated approach, so there is some user input required in order to register the images. And as I sort of alluded to already, the tool of choice, it's not the tool of choice, sorry, if you want to segment hundreds of regions of the brain. It is technically possible to do so, but it would probably be quite a bit slower than alternative approaches. But we still think there's a number of scenarios where you would want to use this. And finally, it's not inherently a quantification tool. However, the design of the software allows you to use quantification from a number of different sources and integrate that into this pipeline quite simply. So the design allows for quantification either by built-in Fiji or ImageJ functions, or alternatively, you can use external programs such as Self-Profiler or Elastic, which is what we commonly use, which is open source trainable classifier type program that you can use to quantify your signal of interest. You can then take the binary output from Elastic and upload that along with your images. And then when you do your registration, you'll get an opportunity to quantify that pre-binarized image. So just to give you a little brief look at what FastMap looks like here, I'll leave it to Dylan to go into more details. But essentially, as I mentioned, the software functions as an ImageJ plugin. You have an uploaded folder of atlas plates that would look something like this, as well as a folder of your actual image files. You, for each image, pick out the atlas plate that matches up most closely with your image. And that will load a set of ROIs that you've predetermined onto your image. There's an automated resizing of that atlas plate that then occurs, and an opportunity to make manual adjustments to the set of ROIs on the atlas if there's any deformation of the tissue or lack of alignment of any of the areas. The atlas alignment itself can be performed on, say, a DAPI channel or something similar, and then that's transferred over to the actual marker of interest, which is in another channel. And that allows you then to quantify whatever your signal is that you're interested in within each of the regions that you've then identified. So I'll give you a couple of examples in cases which we've used this and found it quite useful and simple to perform. So the first here is just a simple case study where we were looking at age-related changes in amyloid plaques and how those were distributed in a 5xFAD model of Alzheimer's disease. So here we were just simply interested in 12 major brain subdivisions, and the goal was to determine whether there were region-specific changes in that amyloid deposition across ages. So we perfused mice at 3, 5, or 10 months of age, and then we used FastMap to determine the regional areas and plaque counts. And by doing that, you can see sort of our output here that we get an increase in plaque distribution with age, and you can see which areas of the brain that's happening most readily in. A second case study that we've included here is examining age-related changes in adult neurogenesis and dentate gyrus functional connectivity. So in this case, we had a handful of regions, so about 3 dozen regions of interest that we had determined a priori. We wanted to see if there were changes in functional connectivity between the dentate gyrus and these different regions of the brain. And we were doing that in the context of context memory retrieval to induce CFOS, as I sort of described at the beginning. And we quantified the CFOS with elastic, segment with FastMap to obtain CFOS positive cell counts per region of interest. And from that, we're able to create these network representations of the memory. And specifically, what we're interested in here is looking at as neurogenesis decreases with age, what happens to the functional connectivity of the dentate gyrus. You can see these functional connections between different regions from the dentate gyrus highlighted in blue. You can see that with age, as neurogenesis decreases, you see refinement of that functional connectivity right down to the point that essentially this then matches up with the structural connectivity between the dentate gyrus and the CA3. And so this gives us a really powerful way to look at how that memory is being stored and the connections or the statistical relationship between different regions of the brain. And this all really arises just from the simple aspect of being able to segment out different regions to look at the cell counts within them. So of course we could do this with a more complex atlas registration tool, but for something like this where we're only really interested in a handful of areas, it's a very simple approach to take to use FastMap to do this sort of analysis. So just to sort of summarize the advantages and limitations of the system, in terms of advantages, FastMap is really advantageous when you're looking to register, as I said, just a few regions or dozens of regions of the brain. And it's really useful when standard atlases don't exist. So this can be the case when you have a particular age of animal that doesn't match up very well with an atlas. That can be difficult then to register on. So that allows you to modify an atlas to match your needs. You might be looking at a different species. So the Allen Brain Atlas is the sort of the most widely used atlas for brain registration in terms of mice. But there really isn't an atlas that matches the quality of the Allen Brain Atlas and that exists for other species in a digital format. So that means that you may have to create an atlas in order to be able to work with other species and other species tissue. And perhaps the atlas that you're interested in or the tissue that you're interested in doesn't match the sectioning plane of the atlas, and so perhaps then this allows you to overcome that difference. This is not just for brains, so it can be used for any tissue type. You can use it on, eye development is something else that we've applied this to recently. You can look at different types of tissue throughout the body. And most importantly, I think the program is simple and free, so available for anybody to use. It has not, we haven't put it out online yet. It's under review. If anybody's interested in it, just send me an email and I'm happy to share it with you right away. The limitations, as I mentioned before, it could be time consuming to use this tool if you're looking at sort of all of the brain regions throughout the brain. And as I mentioned, it's technically possible, but probably not the tool that you want to use for that particular job. And another limitation is that you must be able to identify the regions of interest in order to get an accurate registration. So can you tell the boundaries between different regions? There's different ways to get around this. You can use cell density, specific markers that are expressed in different regions. But ultimately, you have to be able to identify the regions for this to be accurate. And this isn't a limitation of FastMap per se. It's a kind of a limitation across Atlas registration. But we do think that that's something that needs to be considered. So I'm going to turn it over now to Dylan Terstege, who's a PhD candidate in my lab. He is the one who has done the primary amount of work on developing this tool, and he's going to give a demonstration of how to actually use the tool now.
Okay, so I'll just share my screen here. And yeah, so with after having gone through all of our rationale and kind of the advantages and limitations of FastMap, I thought it would be nice to actually just walk through a demonstration of using the tool itself. So here I just have a standard ImageJ download, and I'll just open up our images. So here I have just a sagittal section of an adult mouse brain stained with DAPI. It provides nice contrast for registering images, for registering your regions of interest. Coinciding with that image, I also have binarized CFOS, which was segmented using elastic, and vasculature, which again was also segmented using elastic. So in order to look at the regional densities of those two labels, we need to align this image to an atlas. So the atlas plate from the Allen mouse brain atlas, which most closely aligns with this DAPI channel image, would likely be this one right here. So to turn this static atlas plate image into an atlas amenable to FastMap use, I've gone ahead and traced a few regions of interest at a fairly low level, shown here in red. And then I've saved that as an ROI set within ImageJ. However, if we were to take that ROI set and directly apply it to our DAPI channel image, we'd see that it needs to be transformed along two different axes here. It's not quite right in terms of its X scaling or its Y scaling. So that's where FastMap comes in. So we'll get rid of those ROIs and start up the plugin itself. So going into ImageJ, you can just navigate to the appropriate version of FastMap and start things up. Following the instructions on the screen, we want to choose the parent folder containing both image channels. So if we remember, I had a folder here called images, which contained our DAPI channels as well as our labels. So we want to point to that folder and select. Then choose a folder containing the desired atlas plates. I've done up a full atlas here, which is also included with our plugin, showing that low-level atlas in sagittal plane. So I'll point to this folder as well. Next, it'll just kind of reiterate our selections and then ask us to identify the registration and label subfolders. So by registration subfolder, I mean something with a really nice contrast you can clearly delineate your regions of interest from. So be it a DAPI channel, a propydium iodide channel, or even a really nice autofluorescence. And then our label, let's start off with vasculature. Moving forward, within each of those folders, we really only had the one image. So selecting the range becomes very simple. We're going to start at image 1 and stop at image 1. But then in terms of the analysis type, we have an option here. Volumetric analysis will return our outputs as a function of the percent area within each region the label comprises of, whereas object counts will delineate each specific object and report the number of those objects within a region. So for the vasculature analysis, I'm going to use a volumetric approach. And since this is our first time running the image through, the ROIs have not already been gathered, so we'll just leave this as no. Selecting OK. We want to move this image, which shows kind of all of the plates that I've drawn. We want to move that over to the left side of our, or to the right side of our screen, sorry, just because it's going to be a little busy. There's quite a few images that pop up. We want to be able to see it at all times. So next, we want to draw a rectangle around our tissue section. This will give some sort of indication in terms of what kind of stretch we need to apply to the atlas plates themselves in both an X and Y direction in order to have them align nicely with our target image. So that should be sufficient. We'll click OK. And now looking at our atlas, I would say that our DAPI channel image most closely resembles that in plate number 9. So we'll enter that here. And now just another manual step where we need to group all of these ROIs together so that we can get a size of the ROIs, so that can be applied to the scaling. So we'll select all of the ROIs, navigate through more to or combine, then click add, and then we'll click OK. And now we can just move and adjust each scaled ROI. So here we have the isocortex, aligns quite nicely, but you do notice there are a few areas where things are just a little bit off. So using the shift key, we can add points, clicking on shift and then clicking on the points themselves, or using command or control, depending on your operating system. Mac, it would be command, Windows, control. We can take points away. So that makes that align a little bit more nicely there. Select OK. We'll move through to the next region on our list. So on and so forth through the list. Just align everything nicely here. Again, and at each of these steps, if we notice that something needs a little bit of a tweak, we can do that. Here. Here. And these modifications allow us to not only fit the atlas nicely to our sections, but also kind of make up for some deformities in the tissue, be it from sectioning or just tissue removal. Okay, so now we have everything fitted to our DAPI image. Following the prompts, we are asked if we want to make any final adjustments, and then to save everything, just so that we have records of that particular image, which comes in handy should you decide to change the thresholding of your image detection, or if we're in the situation as we are with this image, where that we have several labels that we can apply through. So to apply this to the FOSS channel, I'll save this as its own ROI set. So more, save, and then we'll just give it a name that's specific to this image, ROI set underscore demo. And then OK, we'll batch through, gather our results. Since we only selected one image, a prompt will come up and say that the data set is complete. So just click OK. And we can open up and kind of look at these results. So since we chose volumetric analysis, you'll see it'll give us a list of our regions, the region area, the label area, as well as the percentage of that region that was labeled. Since we only looked at one image, we only have these regions listed once. However, if we had a second image loaded in, regions would then be appended at the end of that list and populate in here, so on and so forth throughout your data set. So having completed that volumetric analysis, let's go back in and run FastMap one more time to look at our FOSS data. So again, we'll point our macro to the image folder and our atlas plates. We will use DAPI once again as our registration subfolder, and then we will use FOSS as our label subfolder. Again, a single image, so it keeps things simple here, and we'll use object counts this time. And as we have already ran this, this image through, the ROIs have already been gathered and saved. So we can select yes here. Now following the prompts, we can select more, open, and then scroll down to our registration or our atlas plate, which aligns with this section. So ROI set underscore demo, select open, click show all, we can see everything there. And then we can click OK. We can adjust if needed. However, everything is nicely aligned, so we can just move right ahead. Now we'll go through and count and measure throughout. Our data set is complete, and we can check out our outputs. So looking at the regional counts, once again, we have our list of regions. We have our number of CFOS positive cells, and then the region area. So this can also be used to determine the regional density of our markers. So that concludes my demonstration of FastMap. As you can see, it's very simple, allows for a lot of flexibility with free-form transformations at every step throughout the registration process itself. It's nice and flexible with our inputs. These inputs were just a binarized image. And I'd like to thank you all for your time.
Thank you so much, Dylan, and thank you, Jonathan. We now have a few minutes to take some questions. Let's aim at securing a 5-minute break for a biological break for everyone before we start the second session. So the floor is open now for a few questions. Yes, Linda, please go ahead.
Hi guys. Well, thank you so much for developing this simple tool. As you know, Dr. Epp Lab has been helping me adapt this technique at HBI myself, and we all kind of started with using whole brain toolkit together, but quickly realized it's not as semi-automatic as it seemed. And so it's very nice to see how simplified you've made the process. And my question to you guys is, are you in the works of developing it for matching coronal plates to get these finer details? And I see like a use for this, not just for like a whole brain connectomics, like that's great, but even for like something simple, so like illustration purposes for exemplary figures where you have to register for a couple of figures, it's always frustrating to take, you know, the SVG files from the Allen Brain Institute and manipulate, erase like outlines that you don't need because brain never is following like a perfectly linear transformation. So I see multiple use out of that, and like, congratulations.
Yeah, I can answer that. Yeah, so I think that is definitely a use for it, and it would be simple to do that. You would just have to create, I mean, if you're interested in 1 or 2 regions to illustrate, you would just have to basically create an atlas that just has those 2 ROIs in it. and then apply it just like Dylan showed there. In terms of atlases, we have actually created a coronal atlas set, and that's uploaded with the package as well. So we have horizontal, coronal and sagittal atlases at a couple of different levels of structural hierarchy that are included. So that's our starting point. We're happy to share those, of course, with anybody, and we'll continue to share any other atlases that we create along the way. But in addition to that, it's a relatively simple process to create your own atlas plates and add them to it as well. If there's something that somebody wants that isn't there yet, they can certainly do that on their end.
Great, thank you.
That's great. We'll take one more question from Honor.
Hi, Jonathan and Dylan. Great talk. I just wanted to ask a question about FastMap. Does it work on 3D image stacks? Because I noticed in the demo you only tried it on one single image. And if you do that on 3D image stacks, do you have to do the manual scaling in each stack by yourself, since you know how the brain transitions from regions to regions, so each stack would be slightly different in a sense?
Yeah, do you want to handle that, Dylan?
Sure. So so long as the image, as the data set itself can be transformed into a 2D plane, from the
Maybe we
which can then be aligned to atlas plates. What was that? Sorry.
We lost you, Dylan, in the middle. Can you just please quickly recap that?
Oh, sorry. So with the 3D data sets that I've generally been working with, it's mostly been serial 2D images, which we then will run Z-stack projections on, so they align nicely with the Allen Atlas plates. Because at the Allen Atlas, there's a gradation between your plates. So while your light sheet scan might have a resolution of like a 1 micrometer, the Allen Atlas doesn't necessarily line up directly with that. So we'll run those max projections and then align our data sets using that. So we have applied this type of approach to a 3D data set previously. It's just, it requires that kind of that readjustment as you go through.
Perfect. Okay, now that we are running out of time, we'll take a 5 minutes short break and we'll be back for the next exciting session on tissue expansion. We have noticed your questions in the chat and they are all awesome questions, and we would actually like to relay them to the speakers. And I'm quite positive they will respond to us for any queries you may have. So feel free to drop in your questions in the chat. See you all in 5 minutes. Thank you again, Jonathan and Dylan. Very nice to hear this talk.
Thank you.
Hi, everyone. I think we'll get started again. So I wanted to introduce Mathias and Sarah. They're graduate students at the Center for Brain Health. Mathias is supervised by Anne-Marie Craig and Mark Sembrowski, and Sarah is supervised by Frida Miller and Brian McVicker. They both use expanded tissue in their thesis projects, and they'll introduce expansion microscopy before we move on to our guest speakers. So Mathias and Sarah, please go ahead.
Thank you, Jeff. So hi, everyone. My name is Mathias, and I'm going to give you a general overview of the concept behind the expansion microscopy. So initially published by the lab of Professor Edward Boyden in 2015, the idea is to expand homogeneously, isotropically, a tissue, resulting in a physical magnification, which is added to the optical magnification obtained with a microscope. Moreover, during the process, many cellular components are removed, such as lipids, which increase the accessibility of the antigens to the antibody and also make the tissue completely transparent, preventing the scattering of the light and enabling to image deeper in the tissue. With this technology, we can access the organization of tiny subcellular structures. For me, my main interest is synapses, like we can see here. And we can, with this tissue, with regular confocal microscope, we can see the different part of the synapses, the presynaptic part with the active zone, and the postsynaptic part with the postsynaptic density using active zone markers such as RIM and PSD95 for the postsynaptic density. So I can show here the gain of resolution obtained with the expansion microscopy. So on the left, it's a regular tissue, mouse hippocampal tissue, which is processed regularly with PFA fixation and immunostained without expansion. We cannot distinguish the presynaptic part and the postsynaptic part. While with the expansion microscopy, we can see clearly the position, the alignment of the rim in green and the PSD95 in red. And this is obtained with regular confocal microscope without using any super resolution microscope techniques. So just to give a general overview of the workflow behind the expansion microscopy, these methods consist of 4 main steps. First, there is the anchoring of the molecule of interest, which can be proteins, but also nucleotides or lipids. And then the polymerization of a swellable gel to form a hybrid between the tissue and the gel. The denaturation part, which will remove all the lipids and the cellular component and will break the bond, the inter and intra-protein bond. And finally, the expansion of the tissue by incubation into deionized water with movement of water from the medium to the tissue to expand homogeneously the tissue.
Since its initial development 6 years ago, subsequent iterations of the expansion technique have been developed to look at different biomolecules. This slide contains a non-exhaustive list of EXM techniques, including expansion of lipids, ectogenetic modifications, and mRNA. These slides and these papers are listed on our OSF platform. Since its first iteration, different imaging techniques and data analysis software have also been developed and modified for use with expanded and cleared tissues, such as light sheet microscopy. Tune in tomorrow for more discussion around light sheet imaging with expansion and tissue clearing, as well as analysis of these images with our Ariba software. It is my pleasure to introduce our first speaker on expansion microscopy, Marcus Woodworth. Marcus is a PhD student from the Vaughan Lab at the University of Washington. His research interests focus on developing and using super resolution microscopy-based methods to study the chromatin landscape of individual genes within cells. Take it away, Marcus.
Thank you for the introduction, Sarah. Like Sarah mentioned, my name is Marcus Woodworth. I'm part of the Vaughan group at the University of Washington. And I would like to talk to you about this new method we developed called Scepter, in which we are trying to profile the chromatin state of multiple genes at the single cell level by means of expansion microscopy. Now, if you were to look into every individual cell in your body and look at its genetic code, you find that it's really the same code in each cell type. But it's quite fascinating to think that this code can give rise to such a variety of different cell types with their different shapes and functions. This is accomplished by regulating the genes differently throughout this code, so that, let's say, when a progenitor cell is trying to become a neuron cell, just here depicted, it will turn on the transcription factors necessary for that differentiation while repressing any of these factors that would lead to something like a smooth muscle cell. Now, one of the aspects implicated in this gene regulation are post-translational histone modifications. So if we zoom into the genetic code and how it's organized within the nuclei of cells, we'll find that DNA is wrapped around a complex of 4 different histone pairs. Now, these histones, together with the DNA, form the nucleosome as the fundamental unit of chromatin. But each of these histones can have tails that will protrude away from the nucleosome and gain what are post-translational modifications. These are often referred to as histomarks, and the canonically studied ones are acetylation and methylation at lysines of each one of these tails. Now, they are thought to participate in what is the further compaction of chromatin into higher ordered structures about 100 nanometers in size, which could exclude what are factors or other protein complexes necessary for the expression of genes. At the same time, these histomarks are thought to be able to recruit other complexes that might express genes in that general area. In the most compact form, we find chromatin as chromosomes, like what we would observe in a karyotype. But during interphase, this chromatin can be seen throughout the whole nucleus of a cell. When researchers have mapped these histomarks throughout the whole genome of different cell types and compared them between different species, they find that really a combination of these marks seem to either reflect or encode each of the chromatin states that you could find in a genome. For example, at an active promoter region, that is a gene that would be transcribed and expressed, we see what would be the presence of one or all of these different histomarks seen right here. Now, the way that we read this notation would be that histone 3 is here represented as H3. The lysine 4 is that the lysine in position 4 of this histone, and then Me3 would just be trimethylation at this lysine, while AC just says acetylation at the lysine that's being represented here. So these active promoters will have one of these or all of these different histone marks. But at a poised promoter, this is an inactivated gene that could either be turned on or off or expressed at another moment in time. They will have this active mark, the lysine 4 trimethylation, and another mark, which is the trimethylation at lysine 27. So these poised promoters are found a lot in progenitor cells at what would be like a transcription factor. So by removing what is this lysine 27 trimethylation mark, you could then activate the gene. But if you remove the lysine 4 trimethylation, the active mark, then it would be left with a lysine 27 trimethylation, and the gene would be polycomb repressed. Already you see how these different histomarks in combination are really telling us a lot of information of what is happening at each gene. And if you could look at this at the single cell level at every one of the individual nuclei, you can already start telling what is the identity of the cell, what is happening before any gene is being transcribed or expressed, and what that would reflect to, let's say, in some sort of developmental process, or when a cell is being transformed into some sort of disease state like cancer. Unfortunately, the methods that we use to map these histomarks throughout the whole genome have their limitations. The traditional method is a sequencing-based method using an antibody to immunoprecipitate out segments of chromatin. Just to give you a little brief recap of this method, you have right here is the native DNA and the protein associated with this DNA. You will fragment the pieces of DNA throughout the whole nucleus, and then you can use the antibody to just precipitate out what would be a chromatin-associated protein, the DNA linked to this protein, but in this context, it would be the histomark that you're targeting along with the histone and nucleosome bound to this DNA. You can sequence these regions of DNA that you precipitated out, and then just later, after processing the sequencing data, really find where is it that you find these marks throughout a whole genome. This technique has been quite powerful. It's what we've learned so far about where these marks are found in each different genome or cell type. But even as it's been improved up to the single cell level of being able to look at each individual nuclei by itself, this immunoprecipitation really only allows people to look at one modification at a time. And as I showed you before, multiple modifications are really defining these chromatin states. Although there are other techniques that might improve in the number of modifications detected, maybe just here seen on these graphs, like CodeChip or the epitoph, the number of loci detected per cell really decreases dramatically. And there's just this great need for a new method in which you could look at multiple modifications at the same time over a larger number of loci mapped per cell. And that's really what we set out. That's our goal, really, to develop a new method to detect multiple histomarks and genetic loci at the same time and really map out these chromatin states. And we decided on a microscopy-based method because my fluorescence microscopy has really traditionally helped us look at individual cells at the same time and the protein structures within them. Just by using immunofluorescence, you can think about how we can label each individual histomark throughout a nucleus and then determine the density of these marks and regions throughout genomic regions throughout this nucleus. We can also use another technique called fluorescence in situ hybridization in order to detect these genomic positionings by just designing oligonucleotide probes that will hybridize to a specific gene to or even a whole chromosome at the same time if we wanted to, all detectable by fluorescence microscopy. So now you can think about it. Okay, with fish, we know that we could detect the genomic positions and then just look at the fluorescence signal from the immunolabel of each histomark at these fish labeled positions. That would be the principle of how we could determine these chromatin states for every single cell that we want to look at. But there's some limitations that need to be overcome before we really can combine immunofluorescence with fish and establish these chromatin states at each gene. The first one I want to talk about is just resolution. You see here on the left, we have what is a nucleus of a cell that has a K27 trimethylation just immunolabeled. You can see that in this conventional microscope image, at the 200 nanometer resolution or 250 nanometer diffraction limited resolution of this microscope, there's just this haze of signal that we see throughout the whole nucleus. And it just ends up being a challenge to resolve the clusters that are specific to this immunolabeled K27 trimethylation. This group here was able to actually determine where these clusters are found and how this histomark is clustered throughout the nucleus just by using a super resolution technique, in this case being single molecule localization microscopy. You can already tell right here that in these really small clusters, we really could say, hey, there is some sort of gene or genomic position here that might be referring within these K27 trimethylation marks in this image. So just maybe we could apply DNA fish in this case. But the second issue is that there seems to be some challenge in combining what is the immunostain protocol with that for the fish technique. Here is just an experiment that we did where we immunolabeled the same mark you see on the left side right here. This is this repressive K27 trimethylation mark. And we see a couple nuclei for this image. We can really immunolabel it, see it. But once you treat this sample with what is the harsh solvents and temperatures needed in order to denature DNA and introduce the oligonucleotide probes, we see a loss of that immunostain, really necessary for if we wanted to quantify fluorescence signal at each genomic position. You can even do the DNA fish treatment first, and then after immunostain, we found that if you don't do anything to preserve the immunostain, you lose that signal and are unable to quantify the marks at genomic positions. So these are the two challenges that we need to address. That is resolution and some way of preserving the immunostain before or after doing DNA fish. And what we found is that there was one technique that we could use in order to just kill two birds with one stone, and that is with expansion microscopy, which I'm thankful for Sarah and Mathias for introducing. That is really the basis of our new technique, which, like I said, it's we termed it SEPTOR, and that is single cell evaluation of post-translational epigenetic coding. Here is where you can see the acronym spelled out. So how is it that it works? We start out with a fixed cell with a nuclei just being grayed out right here. And this right here is just kind of like a simplified window of the region of interest that we're just trying to, profile the chromatin state. We can immunostain those different histomarks, and we can do more than one histomark by just, changing the different combination of antibodies that we're using to target each mark. And then by applying the expansion microscopy technique, we just link the whole sample to this hydrogel, including the antibodies that we already introduced, and then digest away any rigid structures and then isotopically expand it to give us that resolution we need in order to determine what are those clusters that we had observed before in the previous slide. So that's already solves problem number one about resolution. The second one being that of preserving the immunostain is accomplished at the same time, since we're linking these antibodies to this hydrogel. Therefore, we can follow up the experiment with the DNA fish protocol, submit the gel to would it be harsh solvent or temperature conditions, and introduce these oligonucleotide probes so that we can record the genomic positions. After doing all of that, by carefully, determining each of the genome positions, segmenting them out, we can then quantify the fluorescence signals of the different histomark antibody stains at these positions, and then have this quantitative measurement that we can compare either between alleles of the same gene, between different genes, all in the same nucleus, really providing a lot of information that really, using like sequencing based methods, we can't obtain. That of more than one histomark per nucleus that we're looking at, or just, distinguishing between alleles that have actually the same genetic code that actually isn't as feasible in the case of using some sort of like ChIP-seq or immunoprecipitation technique, like I mentioned to you. So there's still a lot of information that we can glean for this. But we wanted to make sure we had confidence that we could find the corresponding protein structures or, histomarks at specific genomic positions after the whole expansion technique. So we started off first, having some proof of context experiment by looking at centromeres, which is just like these very larger regions that have, centromere associated proteins that we can immunolabel, something that we did here in red, and a specific repetitive alpha satellite DNA regions corresponding to each centromere. So here we have an expanded cell, and now for the rest of the talk, all the scale bars are just going to be represented in pre-expansion units. Here is just, we did a hook stain, or just the whole nucleus in blue. We immunolabeled the centromere associated proteins in red, and using DNA fish, we labeled these alpha satellite repetitive DNA regions in green. And already in this one example cell, you can see this high and strong correspondence between the protein structures and the centromeric DNA within this nucleus, really giving us confidence that we can use this technique in order to look what structures are found at genomic positions. We also did quantify this, by looking at fluorescence signal within just the centromere protein immunostain clustered regions and that of the DNA, something just represented here. All I want to emphasize for the moment is that the distribution of this immunostain signal is pretty close between the two different regions and are dramatically larger compared to if you were to just sample randomly throughout the whole nucleus and look at those signals. But the centromeres isn't what we were wanting to plan. We want to actually target a non-repetitive genetic region, just like anywhere else you'd find genes and things important like transcription factors. We started off targeting what is a housekeeping gene and 2 different histomarks that are, typically studied. And here we have what is a nucleus where we start stained in red an active mark, that K4 trimethylation I mentioned to you before, and in blue the repressive mark, the K27 trimethylation. After this immunostain, after the expansion protocol, we then followed up with a DNA fish labeling of the housekeeping GAPDH, which has to be expressed in all of our cells. So when you zoom into the regions that have these loci, so this of course, we obtain a stack of images. This is in 3D that we're looking at the whole nucleus of each cell that we're imaging. But the image I'm showing right here is just the middle stack. I just happened to find a cell that had both of its the GAPDH alleles in the same plane, which is why I am showing it. But when we zoom into each of the regions that have the GAPDH alleles, you start seeing some interesting facts. One, this housekeeping gene, which we know has to be transcribed, you do not see any of the blue signal or the blue clusters within the GAPDH loci regions, just by looking at the images. But you do see that at least for one of them, there is some overlap or presence of fluorescent signal for the active mark, the red one, at these green GAPDH loci. But it's not the same between the 2 alleles of this nucleus. And one has a lot more higher levels compared to the other one, really showing you the heterogeneity of the states that might be present for just an individual gene. We know it has to be transcribed. We actually, have transcription data from this cell line. This is a human cell line. But that doesn't necessarily translate to saying that they're all going to have the same amount of histone marks for like an active marker. All of this heterogeneity, which is typically difficult to detect in other sequencing-based methods. So what we did is, okay, we want to quantify these levels. We can then use imaging processing and analysis in order to determine the clusters for the active mark seen in here, the repressive mark in blue, and for each one of the different GAPDH loci, along with just randomly sampling throughout the whole nucleus as some way of comparing it as a negative control. This just being the same locus seen in B for this example. So when we do this quantification and we look at the distribution of these loci, here on top is just for the active mark fluorescent signal, and on the bottom is for the repressive mark signal. You see that the representative red clusters, active clusters, definitely have a high level of this active mark fluorescence, which is a lot higher than that if you just randomly sample throughout the whole nucleus. But when you look at the GAPDH distribution in this violin plot, you see that the loci that we were able to detect, they aren't all in the active mark distribution like area. In fact, a lot of these different marks seem to have little to no density of these K4 trimethylation marks. Again, that heterogeneity that I think was quite surprising to us. What was not surprising is that when you look at the distribution of the repressive mark at GAPDH, this one just seems to be a lot like what we expect for random, not having much of a presence of this K27 trimethylation repressed signal. Again, it's GAPDH. We need to have it expressed in this cell line. Now, to really, validate this method, we actually repeated the targeting of these different markers, but now using a sequencing-based method like I had introduced to you before. In this case, now we're just looking at the number of tracks or reads that we obtained in a whole population of cells that have a K4 trimethylation mark versus K27 trimethylation. So here, just is we're talking about the genome positioning. In this highlighted region was where we actually have the FISH probes. So we were wanting to see which of these regions have either K4 trimethylation or K27 trimethylation. And when you compare the top to the bottom tracks, you see that there is these traces and peaks that correspond to K4 trimethylation, indicating the presence of it in this GAPDH region, but really an absence of these same peaks in the K27 trimethylation mark. All to say that, Scepter really is capable of quantifying and detecting what is the differences in histone marks at this housekeeping gene. And so I'll continue on with what is going to be two other examples. And then I just want to, take some time to go through what would be the workflow. So the next example is just, we can target more than one gene in each cell. So this really is just a cell where we targeted in magenta what is the active mark, and we label at the same time 3 different genomic regions, that for MYL6, HOXA, and LINC-PINT. We already know MYL6 and LINC-PINT are actually transcribed, but HOXA is repressed, being a developmentally regulated gene responsible for limb development. When we look at either the distributions or even the image themselves, you can definitely see the presence of the active mark at the MYL6 and LINC-PINT regions, but not at the HOXA, which you see for a whole population of loci. This is just right here. I know there's a lot of information on the bottom, but focusing on the highlighted regions where you will find the MYL6 or the HOXA cluster or LINC-PINT, you see that, again, the one on the left and the right have the presence of the active mark, but HOXA doesn't. So really showing and validating that Scepter can detect histone mark levels across multiple regions in the same cell. And the second example is just that, you don't have to necessarily focus on histone marks. You can also look at other structures within the cell. So here is just a loci example for, in red, the active mark clusters, in blue what is this transcription initiation marker, and in green the GAPDH, like I showed you before. In this example, it's quite interesting that if you focus in where the locus is, you see the presence of both the transcription initiation marker and the active marker at the same time. In both of these alleles, this is actually from the same cell. When you look at the correlation of K4 trimethylation signal versus POL2 S5P, this transcription initiation, for every one of the green loci, this is the GAPDH loci, across all the cells that we looked at, you see the strong correlation between these two different levels. Really showing this, there may be some connection between what is this active mark and the transcription initiation, something that has been, hypothesized before, but now we're looking at it at the single genetic loci level. Now, because of the fact that this is a workshop that we're talking about, and I'm sorry, and it's unfortunate that we can't, do some hands-on demonstrations. I just want to show you what would be the workflow of how we work with this Scepter protocol, where first you would want to select the genes. You actually have to design the probes for every individual gene that you're interested in targeting. It would be nice if you consult maybe some ChIP-seq data from the sequencing based methods to see which ones have specific marks that you want to target. And from those genes that you selected, you design those probes. And after you have fixed and have the sample that you want, you would immunostain the histone marks within that sample, use the expansion protocol to look at the higher resolution and preserve that immunostain, then do the DNA FISH to then look where in each one of those genomic positions you see this fluorescent signal. And I focus mostly on just quantifying that fluorescent signal, but you could also think about it as just a distance and proximity to what would be like a repressive cluster or a active cluster, or maybe just the overlap of each genomic position with the immunostain cluster determined clusters. Then you would image, you would take 3D stacks using a confocal microscope, and then process these images and analyze them in order to establish what is the profile for every individual either locus or nucleus. So you can draw these comparisons about between histomarks in each region, or between, you know, alleles of the same gene, or different genes. Now, all of this, we actually decided to make a step-by-step protocol, which if you go to protocols.io, you would be able to find it for this scepter method. You can also go to the Von Group website where, you know, in our publications page, you would be able to easily also find this protocols.io, because we want to be able to, you know, share this information with you, and you apply it into what you want to be able to profile. Now, just to recap everything, this scepter method can quantify multiple histone marks within each individual cell at multiple genetic loci, which allows us to then compare and contrast the histomark levels between genes, between alleles, or even at the individual genetic loci. So I'd like to thank the people in my own lab, particularly one of the former members, Aaron Halpern, who helped in this project. He's now a research scientist at Harvard. Joshua Wan, the head of the lab, and then the Quay group, who also participated in this project, and the funding that allowed me to do this work in graduate school, both from the molecular biophysics training program at UW and this F31 that I was able to get from the NHLBI. Are there any questions?
Dr. Sienia, go ahead.
Hi, yes, that was amazing. Thank you for sharing. Super cool work. And I wonder if you have a feel for what happens to the DNA and the histones when you do the expansion. Does the DNA actually expand, or are you increasing the space between the histones, and can you preserve like heterochromatin versus open chromatin? And have you tried any of this with DNA methylation or non-histone marks that you might want to look at?
Those are all great questions. There's a lot to unpack from that. Well, so we do definitely see, like, you know, we can see that at the macro scale that the whole nucleus is still preserved. And as long as you're being careful of the whole expansion process, at least on the macro scale, we see a nice preservation of the nucleus. Now, if we're talking about at the nanometer scale, in which there might be some slight shifts, we don't have the resolution to be able to detect it. All we can say is we did try to do, like, for example, in that experiment with the centromeres, where we did try to at least map the interaction between the protein structure and the genetic regions together. And we also done correlation to make sure that, you know, even on the protein structure, that when we compare before and after expansion, there seems to be some great correspondence in that case. So I do think that definitely we are interested in wondering if maybe heterochromatin, the preservation is happening differently than what would happen in like euchromatin. Now, in terms of what's happening within the gel, we do know that proteins will be able to be linked to this hydrogel, including what is the antibodies. The DNA itself might have some sort of linkage. There's still strong interactions, like the whole nucleosome is, we think would probably preserve pretty well in these expanded gels. But just also the entanglement of these polyacrylamide chains throughout the whole nucleus, we think also plays a role in making sure it's all expanding. But again, we don't have the resolution to be able to say maybe at the nanometer scale. All we can say is that we are able to obtain interesting information about these chromatin states using this method.
All right. Thank you very much, Marcus, for a fantastic talk. If there's any more questions, feel free to put them in the chat. And I'm sure Marcus can answer them during the talks. Next up, it is my pleasure to introduce our next speaker, Dr. Edward Boyden. Dr. Boyden, I believe you are on. There you are.
Hi, everybody.
Dr. Boyden is a professor in neurotechnology at MIT, an investigator of the Howard Hughes Medical Institute and MIT McGavran Institute, and professor of brain and cognitive sciences, media arts and sciences, and biological engineering at MIT. Dr. Boyden also leads the synthetic neurobiology group, which develops tools for analyzing and repairing complex biological systems. Today, Dr. Boyden will be discussing the development and applications of expansion microscopy, and we are so excited to have you here. Feel free to take it away.
Great, thank you. My PowerPoint, for some reason, is saving very slowly, maybe because of all the pictures and movies and things. So I apologize, I can't press the show screen button. But yeah, so you've already heard a bit about expansion microscopy already. I guess I'll just very briefly introduce it then and not spend a lot of time on it, and then jump right to the newer things that we're doing that are either recently published or unpublished that might be interesting to contemplate. All right, great. The PowerPoint is coming back to life. So let me just do screen share. Oops, what just happened to it? Sorry. Okay. All right, so let's start the. All right, and now the screen sharing will begin. They moved the share screen button. I keep changing Zoom. Okay, there we go. All right. Great. So yeah, thank you so much for the kind invitation to come and talk. Our group has been working a lot on ways of trying to democratize nanoimaging. And one of the key strategies that we've been putting forth is this idea that we can physically magnify biological specimens by synthesizing dense spiderweb-like, heavily cross-linked hydrogels made out of swallowable polymer that densely and evenly permeate biological specimens. Sodium polyacrylate winding its way inside cells and outside cells, in between biomolecules and around biomolecules. And the core concept really has a few different parts, but the goal is to be able to take some kind of cell like the one on the left of this cartoon and to pull the biomolecules apart from each other. So ideally, two biomolecules that were touching are now some minimum distance apart. Two biomolecules that are some distance apart will be pulled apart by a linear scale factor. And of course, the goal is that we can then do nanoimaging on a conventional microscopy setup, because the expansion process ideally is isotropic, or preserving of the organization of the biomolecules. So in an ideal world, you'd have a constellation of biomolecules hovering in space in 3D with their relative organization preserved. So expansion really boils down to a couple key steps. In this cartoon, you can see one of them, which is to create anchors that will bind to different biomolecules, whether proteins, nucleic acids, whatnot. We're working on ways of expanding, sugar and lipid labels and all sorts of other things now. Then, of course, you have to create the polymer network. And in the original protocols, what's done is to do free radical chain growth polymerization, where in this case, in the cartoon, little white spheres representing sodium acrylate will form these sodium polyacrylate gels. And then when they encounter a little handle that is bound to a biomolecule, here shown in purple, they'll form a covalent bond. At that point, some kind of softening step occurs. You can destroy molecules you don't need. You can also soften them through temperature and heat application or detergent. And then you add water, and then the polymer threads will expand away from each other, and you'll get a physical magnification of the object. So we first reported that this was possible in 2015. Panel B shows a small piece of the mouse brain. Panel C shows the same piece of mouse brain about a day and a half later, and it's been expanded by about 100 times in volume, roughly 4.5-fold in each dimension. And the polymer network starts out quite dense, such as in the sketch in the upper left. The mesh size or polymer spacing has been found to be of similar hydrogel networks, has been found to be a couple nanometers using small angle neutron scattering and other technologies, about the size of a biomolecule or smaller than many biomolecules. And after you expand, you get an extended conformation in the lower left. So when we first announced this, of course, one of the big questions is, well, is expansion even? And so by design, we want it to be very even. The density of the polymer, the caustic nature of it, the hyperswallability and the physics of it, which other people more versed in polymer physics than I have worked out over many decades. And then, of course, the softening of the tissue. In the early days, we used proteases, but now many forms of expansion strategies exist, and you can use proteases, heat, detergent, and all sorts of methods, as I alluded to earlier. But of course, we wanted to make sure that this was actually true and not just an assumption. And so in the early days, and continuing to this day, we spent a lot of time taking pre-expansion images using a classical nanoimaging method like SIM or STORM, and more recently in some newer work that we haven't yet published, DNA Paint. And now we're also starting to explore STED a little bit. And then after we expand, we can take a picture of the same specimen with a regular diffraction limited microscope. And so the distortion is not zero, but it's pretty small. We get distortions of around a few percent over a typical microscope field of view. And so that's quite acceptable for the vast majority of biological applications. Regarding resolution, the fundamental resolution of the hydrogel, of course, is determined by the inhomogeneities of the hydrogel, as well as the labeling density that's used. But we estimated resolution in the early days by taking pictures of known things like microtubules, decorate them with fluorescent antibodies. You can take a pre-expansion image and a post-expansion image. The post-expansion image, we can then deconvolve or divide what we see by the known ground truth, decorated with antibodies, of course. In the case of microtubules, we know a lot about what they should look like. And so we found that to first order, the resolution was the resolution of your microscope divided by the expansion factor, which is a nice clue, right? That means as you expand more and more, you might be able to get better and better resolution until you hit the inhomogeneities of the hydrogel or the hydrogel's expansion process. So this basic idea, anchor key biomolecules or labels to the a polymer network that you synthesize densely and evenly, soften the specimen and expand. Many groups, including some of the speakers around this virtual table that we're all at, have developed important variants of this basic strategy. And the number of experimental preprints and publications, not review papers or whatnot, but the number of experimental results is really growing by leaps and bounds. I think the number of experimental papers and preprints is about to hit 300 any day now. I think it's at 292 at last count. And people are expanding all sorts of stuff: bacteria, the human kidney, heart cells, fruit fly brains, and the list goes on and on and on. Importantly, many people develop their own versions of expansion that are customized for particular problems. So for example, when people have been expanding bacteria, they might add an extra enzyme to soften the bacterial cell wall. Or when people are in our group for expanding C. elegans, which has a very tough collagen-equipped cuticle, we added a collagenase step to help confront that head on. And so it's really exciting to see an expansion community growing up where people are physically magnifying all sorts of stuff and often cleverly optimizing key steps in order to make expansion optimal for a given question. For protocols from our group, they're on our website. We have a website, expansionmicroscopy.org, which contains protocols that our group has developed or helped develop. And we also used to help host lots of workshops and host in-person visitors. With COVID, of course, we haven't been able to do that. But last summer, we posted a little tutorial with step-by-step procedures, including how to handle these delicate, sometimes fragile samples, although they don't have to be delicate or fragile if you treat them properly, and to manipulate them so they can be properly imaged. So that's kind of a quick, review, the first couple of years of expansion. And so, yeah, I'd love to focus the rest of my talk on some of the newer things that we've been doing. And one thing that we've been really excited about is that expansion might not just enable nanoimaging on regular microscopes, which is now, as I mentioned, kind of widely appreciated with literally hundreds of experimental papers and preprints doing such things, but can we also separate molecules for better labeling? And so I'm going to mention some work here by Deblina Sarkar, Jinyun Kang, and Oz Wassi, with great help from Margaret Schroeder in our group, and Amuchi Ebenari and Christina Kitko have also been developing important variants of this technique. And the basic concept is, what if we could separate proteins from each other and make them far enough apart that then you could introduce antibodies that could then access previously inaccessible and therefore invisible, as far as we're concerned from a light microscopy standpoint, epitopes. So most expansion studies are expanding around 4.5 to 5-fold. A couple groups have presented 10-fold expansion protocols. And our group a couple years ago presented a prototype of a 20-fold expansion protocol, although it required end users to do some chemical synthesis of their own. And so it hasn't really been used very much, if at all. So what Deblina, Jinyun, Oz, and others have been doing in our group recently is to work out a 20-fold expansion protocol that uses all off-the-shelf chemicals. And also, as I might note, has this added perk of being able to decrowd proteins from each other. So this is shown in the cartoon at the top. Here's a synapse jam-packed with proteins, of course. And if you label with fluorescent antibodies, you can see in the upper right of the cartoon a red and a green antibody labeling proteins on the outside of a densely packed cluster. But if you expand 20-fold, and we do this by expanding twice, basically. We expand once by 4.5-fold for a second polymer in the space opened up by the first expansion, and we expand it again. So the second row of the cartoon, you can see now that the yellow and blue antibodies can access proteins in the inside of this complex, whereas before they could not. So we wondered, could we use this to make new biological discoveries? And so we collaborated with Tom Lampine's group at the University of Maryland and Li Wei Sai's group at MIT, experts on synaptic nanostructures and Alzheimer's disease, respectively. And we started asking the question, could we start to see new biology that's difficult or sometimes even impossible to see with earlier techniques? The first thing that we did, and we're still working on the figures for it, in fact, we're just discussing the analyses earlier today, is to do the pre-expansion followed by post-expansion analysis of how much distortion were introduced. So I apologize, I don't have the figures for you. But if we do DNA paint, a very good super resolution technique before we expand, and then we look at the same samples, same labels after we expand, we can calculate how much distortion is introduced by the expansion process. And so it's pretty low. We were just analyzing the data today, but it could be as low as 5 to 15 nanometers of error, which is quite good. These are some pictures of synapses. At the bottom of the slide, the different antibodies are labeled by the color code. And so we're very excited that you can see really detailed nanostructures within synapses. Okay, can we decrowd proteins from each other and see previously invisible things? Well, in panels B, C, and D, it's kind of a crowded slide, but let me walk you through it. Yellow is pre-expansion staining and purple is post-expansion staining of the very same specimen with the very same antibody. And so in panel B, you can see a calcium channel that is invisible before we stain. You don't see much yellow, but after we expand, you can see calcium channels at these synapses. And we know they're synapses because we have reference stains that don't change very much when we expand. Panels C and D show other pre and postsynaptic proteins that are vastly brighter when we stain post-expansion than pre. Not everything is crowded. Panels F, G, and H show 3 different proteins where the pre and post-expansion staining look quite similar: Homer 1, Bassoon, and Shank 3. So working with Tom's group that I mentioned earlier, we asked, well, now that we can see these calcium channels that were not visible before when we stained before expansion, can we understand something about the nanostructures of synapses? And Tom's group had previously published a nice paper in Nature on nanocolumns, where pre and postsynaptic proteins are mysteriously aligned with each other. I'm not an expert in the biology here, so apologies if I'm butchering either terminology. I'm sure many people on this call are much more well-versed in it than me. But what's interesting here is that we saw alignment of the presynaptic calcium channels with other presynaptic and postsynaptic proteins. So there's this nanoscale alignment across the synapse of different pre and postsynaptic proteins that we can now see because the decrowding enables both nanoscale resolution imaging in intact brain tissues. This is in the cortex of the mouse. So we're able to do this in thick 3D tissues as well, which is a nice perk. and derive nanoscale information about how the proteins are coordinated across synapses. With Liwei Cai's group, we asked whether, of course, densely packed proteins are very noticeable in many neurodegenerative conditions. And so we started wondering, well, amyloid plaques and Alzheimer's have been quite under study, but also a bit controversial. Could we start to image them better and maybe unveil new hypotheses of what's going on? So we just started using these 5x FAD mice, a mouse model in Alzheimer's, and did the same thing, pre-expansion staining in yellow, post-expansion staining with the same antibodies, same specimens, in this case using an antibody against A-beta 42. And what you can see here is that the post-expansion staining shows a lot more detail than the pre-expansion staining. In fact, we even saw these little amyloid dots that you see at the bottom of the slide that were pretty much invisible when we did pre-expansion staining. So it's quite possible that by physically magnifying these biological specimens, we can start to really see nanostructures that are difficult to see, if not impossible, using pre-expansion staining. We saw this with 2 different antibodies against A-beta 42, and we never saw them in wild type mice. This is kind of a crowded slide, but we started wondering, these amyloid dots that we started to see with post-expansion staining, are there any other interesting biomolecules contained within them? And interestingly, these amyloid dots seem to have a core inside of them that contain interesting things like potassium channel subunits. So here in purple is A-beta 42 being labeled post-expansion. In yellow is a potassium channel subunit, and they formed little clusters that seem to be similarly periodic with the A-beta clusters. You can see here in panel D, a Fourier transform that shows that they have a separation of maybe a couple hundred nanometers. But they had interesting geometries too. We're still analyzing all the data, but our hope is that we can start to understand a bit of the biology of what's going on with these A-beta puncta and the KV 7.2. That's the potassium channel subunit puncta that are localized within. So short story number 1 to conclude is that expansion microscopy, which is already quite widespread for enabling nanoimaging on conventional microscopes and is pretty streamlined and easy to use, there might be other applications of it, such as decrowding proteins from each other in order to yield insights into dense nanoclusters, dense protein packed organelles, dense protein environments like synapses, and other biologically important nanomachines. Now, another story I want to tell you is about how good can the expansion get? Can we really get down to single molecules, for example? And so Ray Gao and Jay Yu in our group, along with the help of many others, we collaborate with Rachel Neve and others on the scientific side of this. We asked whether different ideas from polymer chemistry could be brought into the expansion repertoire. And the upper left shows one of the things we're really trying to deal with, which is, well, the free radical chain growth polymers, which is what are used in expansion microscopy currently to form these long sodium polyacrylate chains, have different kinds of inhomogeneities. You can see different levels of cross-linking. There are free ends that dangle. There are loops that don't go anywhere. Now, for many years, I think well over a decade at this point, in polymer chemistry, people have been studying these diamond lattice hydrogels. They're shown in the cartoon at the right-hand side of the slide. And they're known for being extremely homogeneous, relatively defect-free compared to other kinds of polymers, although this is a very rapidly evolving field, and many, many people, of course, are trying to build polymers with exquisite homogeneity. So we wondered if you could make these diamond lattice hydrogels with polyacrylate tetrahedra, and to do them, in this sort of step growth process by using click chemistry, for example, to assemble them into a diamond lattice, would that allow us to be more isotropic in our expansion capability than the classical chain growth polyacrylate methods? So that was the idea. And we made different click chemistry strategies for doing so, including some that are deliberately sort of not the most efficient click chemistries, because you want the monomers to saturate and permeate evenly the specimen before they start forming the polymer network. So we actually use some click chemistry strategies that are known to be kind of slow because we want the monomers to have time to permeate evenly before they start forming the network. So I alluded to this idea that you could use objects of known geometry like microtubules to try to probe the fundamental limits of resolution. And so, in this case, we collaborated with Rachel Neve to look at herpes simplex virus. In panel C, you can see on the left some herpes simplex viruses where we labeled the envelope proteins just non-specifically, using the shape of the virus as sort of nature's scale bar, so to speak. And with the tetragel on the left of panel C, we got a very even expansion. So you can see these round shapes. On the right side of panel C is with the polyacrylate free radical synthesized hydrogel. You can make out shapes of viruses, but it's a bit more noisy in the envelope. And the population data is shown in panel G. With the free radical synthesized hydrogels, abbreviated PAAG on the right-hand side, we had a median error of around 14, 15 nanometers or so. But with the tetragel, we actually got down to 9 nanometers of error. And Paulo Kusuri and Xiaowei Zhuang just published a paper a couple of weeks ago where they use these tetragels to expand DNA origami, another way to build sort of a natural scale bar to get down to the fundamental limits of resolution. And they came up with similar numbers, maybe even a little bit better than ours, where the polyacrylate hydrogel had that's free radical synthesized had errors in the tens of nanometers, and the tetrahedral hydrogel had errors down in the single-digit nanometers. So it's quite interesting to think that with some more optimization, it might be possible to really get down to single molecule precision, which I think could be really cool. If anybody could do single molecule resolution imaging with a regular old microscope, this gets quite exciting. Now, why do you care about the shapes of the viruses? Well, I'm not a virus expert, but we created a couple different. We didn't create them. We adapted a couple different metrics of shape, including the ellipticity and the sphericity and so forth, and showed that these were much better preserved with the tetrahedral hydrogels than when we used the classical free radical hydrogels. So short story number 2, it's very exciting to think about whether innovations from polymer chemistry might, in the coming few years, be broadened increasingly into the expansion community and allow us to get down to single molecules, which would get very, very exciting because, of course, if you really can map out molecules and how they're organized down to single molecule precision, inferences about potential interactions between proteins and so forth might be very exciting. Now, of course, no microscopy technique can fully prove an interaction between two things, right? Even two things that look like they're touching might not be interacting in any functional way. But these strategies in microscopy could be used to generate powerful hypotheses that then could be tested through classical biological perturbations. Okay, so that's the short story number 2. And then finally, I would like to talk a bit about multiplexing capacity. And then lots of people have been adapting different ways of multiplexing through multiplex antibody staining, multiplex RNA imaging, and other methods into the expansion toolbox. I'll just tell one short story here, which is a massive collaboration between many, many groups. But we worked with Aviv Regev, George Church, and others on this. And that's the idea of bringing nucleic acid multiplexing into the expansion context. So expand a specimen, and then you could, after you expand, do some kind of serial process in order to read out a lot of information from biological specimen. The version of this that I'd like to talk about is the one that at this point is the most reliable in our hands because it has the highest yield. And I should mention this is work that was quintuply spearheaded by Shahar Lon, Dan Goodwin, and Nisinha Oswasi and Fei Chen. So his basic idea is let's anchor proteins, RNAs, other things, form the hydrogel and expand them away from each other. And then we can bring in different probes that have interesting properties and then read them out through some kind of sequential process. In this case, we use the padlock probes that bind to RNA. These are small pieces of DNA that hybridize to their target and form these padlock shapes, hence the name. We ligate the padlock into a circle using an enzyme. We amplify the circle using rolling circle amplification with a simple polymerase that amplifies the sequence. And then we can sequence a small barcode on the other side of the padlock probe. And when you sequence a nucleic acid, you're basically copying it. And as you copy it, you're adding fluorescent nucleotides. So you blink out the identity base by base under the microscope. So we call this targeted expansion sequencing. And we thought to try to apply this to the mouse visual cortex shown in the upper right. The Allen Institute has been working on the visual cortex for a while, doing a lot of single cell RNA-seq. And so we collaborated with them and we developed a couple dozen probes, these padlock probes to bind to a couple dozen targets that they had identified. And clusters of cells appeared when we looked at them, as shown in the upper left with the classical clustering analysis. In the upper right is the mouse visual cortex, layer 1 on the right, layer 6 on the left, and the color code of the upper left, the clusters, has now been superimposed over the layers of the cortex. You can really read out the cell types and where they are located. Now, there are many other ways of doing that. Mehwish, for example, has been applied to look at cell types of the mammalian brain, amongst others, from Zhaowei Zhuang's group. But with expansion, you can start to look at very fine compartments of cells. And so building from some gene lists identified by Aaron Schumann's group and others, we wondered, could we really hunt down transcripts, RNAs, in individual synapses, dendritic spines, and other small compartments of brain cells in the intact hippocampus of the mouse? So we expanded, developed a set of probes for 34 genes that, again, Aaron's group and other groups have pinpointed to be on dendrites of hippocampal and other neurons. And we then did this targeted expansion sequencing of the barcodes on those probes, as I told you earlier. And we were able to map out the location with nanoscale precision in individual dendritic spines of these messenger RNAs. So you can see some nice pictures of that in the lower left. The gene names are shown in green. But perhaps most interestingly, in the lower right, we can really map out where genes were located in spines, dendrites, and other compartments and quantify them. And so there's some, I hope you can see my mouse, there's some transcripts that are found in spines of CA1 pyramidal neurons, but they weren't found on the dendrites that were just, of course, nanometers away, because, of course, the spines are connected to dendrites. So there can be drastically different localizations of RNAs in different compartments, separated by very, very tiny distances. One thing I really was excited about was we looked at different cell types. We looked at dentate gyrus neurons and CA1 pyramidal cells and found similar distributions of RNAs along their dendrites. So there might be more general rules about how RNAs are expressed in different parts of brain cells that connect up with maybe underlying principles that we don't even know about that govern how RNAs are transported in different cell types in the intact brain. Working with Avi Reger's group, she gave us some tumor samples to look at, and we started mapping out. She helped develop a 300 gene panel, and we started looking at different immune cells and tumor cells in a. This is actually a breast cancer metastasis to the liver from a specific patient, de-identified. She just gave us the specimen to look at, part of this human tumor atlas network. And as you can see in panel D, we saw lots of colocalizations of different immune cells with each other, different tumor cells with different immune cells, and so on and so forth. So there's a lot to explore there, but I'll just give you two examples shown in the lower left and the lower right, which were pretty striking. So I think many of us have heard of tumor immunotherapy. Tumor cells and immune cells interact, and if you could interfere with that, you might help treat patients, and that's been very successful. Won a Nobel Prize for some of the inventors of it. But if you look at the lower left here, some B cells are shown in cyan, and some tumor cells are shown in yellow. And as the closer the B cell, an immune cell, was to the tumor cell, the higher the expression of an interesting marker that's implicated in tumor biology. And I'm not an expert in tumor biology, but we're very excited to see that you can map out tumor immune relationships that might have interesting interaction implications to be tested with further science. In the lower right, we saw that tumor cells, when they were close to a certain class of fibroblast, vastly upregulated a specific hypoxia-related factor. And of course, tumors and their ability to resist low oxygen has been of great importance as well in the study of tumor biology and the pointing towards different therapeutic avenues. And so now you can map that out very efficiently in tiny tumor cells. So to summarize, we and many others have been exploring different multiplexing strategies to be brought into the expansion arena. And we're very excited about where this is going to head in the time to come. But I guess, it's been really exciting to see what others have been doing as well. And maybe I'll just end on this slide, which is text too dense to read. Just to say, it's really great to see an amazing community spring up, adapting expansion for many, many different purposes, optimizing it for better resolution, higher expansion factors, different specimens that are tough, like bacteria and C. elegans and so forth. And really to see a lot of beautiful crosstalk between different groups that's resulting in an exciting, powerful, and really democratizing of nanoimaging toolbox that's spreading very quickly. And so it's really great to be part of this community with you all. I think I'll end there. I think I acknowledge on the way all the people who helped with these projects, and this is actually the acknowledgement slide for our group's entire body of work, which of course includes other tools like optogenetic tools and so forth. So not everybody on this slide worked on expansion, but it's just a very powerful message of how this is not just an interdisciplinary field anymore. It's, you might call omnidisciplinary. Every field in science is almost colliding with this quest to try to help us understand what's going on in complex biological systems. And so I'll just end on this slide, acknowledging at the top of the slide our wonderful group members and alumni who've helped with these projects and many, many others. And in the middle of the slide, an even longer list of people all over the world who we've had the privilege to work with. And as a final note, our website, syntheno.org, you can find protocols for all of these techniques that I've talked about. And in the case of custom reagents that we've had chemistry companies make, how to acquire new reagents such as the tetrahedral hydrogels, which required custom synthesis. And with that, I'd be happy to take questions.
Thank you very much, Dr. Boyden, for what I can say was a fantastic talk. We have one question in the chat from Roger. It says, great talk, Dr. Boyden. One, does expansion break up some protein-protein interactions? And question two, at the highest resolution, 2,200 nanometers 20-fold is 10 nanometers. If accurate, co-IF an issue as the fluorescence tags are relatively away from the actual epitope, antibodies potentially pointing away from each other.
So to the first question, a 20-fold expansion with a 300 nanometer typical confocal means a resolution of around 15 nanometers or so. So it's not quite sufficient to see whether most proteins have been separated or still touching. Our hypothesis, because when we look at the amyloid plaques expanding or the synapse expanding, is that we are breaking up proteins from each other and separating them. So two proteins that were touching are now some minimum distance apart, and two proteins that are some distance apart are scaled up by a linear factor. But to be honest, I don't know if any expansion protocol has enough resolution to prove that two proteins that were touching are now some very small distance apart. That's an exciting direction, and maybe some of these new polymers will get to the resolutions required. As far as the resolution, if you expand first and then apply the antibodies later, that problem mostly goes away. You know, if you think about it, if you expand all the proteins away from each other and then deliver the antibodies, the size of the antibody is as if you were dividing it by the expansion factor. I mean, not in real life, but virtually, right? So if you have a bunch of proteins that were really close and now they're very far apart, now you bring in antibodies that, you know, like Alice in Wonderland, look really tiny compared to all the distances that it has to navigate, then the antibody error really is reduced. Now, again, we haven't really made any big public claims of that because, again, you know, these are extremely tiny resolutions we're talking about. And just now the polymers are getting down to the point where single-digit nanometer errors can be claimed, but nobody, to my knowledge, has really quantitatively evaluated the antibody error at the single-digit nanometer level. I mean, that's hopefully something that will be done quite soon. Yeah, great questions. Thank you. Other questions or thoughts?
Anyone else have any questions? You can jump on in.
Can I pick up on my own question? If nobody else wants to go. I'm just curious if the chemistry used has a potential to break up natural interactions. So if we want to do a co-IP, or sorry, a co-IF experiment, are we going to miss it because of the chemistry?
Yeah, I don't know the answer to that. I think if one is doing actually, can you tell me what co-IF is? I don't know the acronym, I'm afraid.
So co-localization, basically, co-immunofluorescence.
Oh, yeah.
So are they really physically? As you know, if people have used this as a proof of concept that the proteins truly interact because they're at the same place, but there's an issue of resolution. But if the chemicals perturb that interaction, we may miss it.
Well, I mean, doing co-IF with a regular confocal microscope, which has a 300 nanometer or so resolution, all it tells you is that the proteins are within 300 nanometers of each other, right? So I don't know if that can be called proving an interaction, with all due respect.
No, but they do co-IP. It's not the only tool, right?
Okay.
Co-immunoprecipitation.
Okay. Yeah, co-IP, but then you don't need microscopy at all, right? I'm not an expert in that.
No.
Yeah. So for co-IP,
parallel tools. That's all I'm saying. And together, they tend to prove that the two molecules truly interact.
Great. Yeah, so when you expand, it gives you better resolution, and you would pull proteins apart. But again, the hypothesis, but if you expand something, suppose two proteins were 1 nanometer apart, right? And you expand 20-fold. Well, now they're 20 nanometers apart, right? They're still way close within the diffraction limit of your microscope. So they'll still look completely like they're overlapping. So it will give you just as much information about overlap as a regular CoIF would, but you have better resolution to tell you where things are.
Okay, thanks.
Because the blurring of the microscope really smears everything out, right?
Oh, it's a mess, yes.
Yeah. Now, as we get down to single-digit nanometer resolution, that's where things get really weird and exciting. And that's why I haven't made any public claims there about colocalization implying interaction. But yes, if we, the paper that Pallav and Zhao Wei published that I mentioned uses our tetrahedral hydrogel with LeDue origami, and one of their figures I think claims down to 5 nanometer resolution. So that might be the, if I remember, of course it's LeDue origami, which is a very admittedly artificial thing to expand, right? A protein that's tightly binding another thing might interact very differently. So, and that paper just came out a few weeks ago. So, yeah, this is kind of the frontier right now. Yeah, if you can really get down to single molecule resolution and interactions is extremely tantalizing, but nobody has, I think, publicly claimed a proof of that yet.
Dr. Murphy, did you have a question?
Yeah, fantastic talk, really inspiring.
Good to see you.
I think if I got it right, in some cases, antibody penetration is improved by expansion because we're, I guess, reducing the density of protein. So is this going to allow us to kind of re-examine, I don't know, some hypothesis looking at plaques? Would you also expect to look at tangles with expanded tissues?
Yeah. So ever since we posted that on bioRxiv, we've been approached by a lot of people interested in looking at all sorts of densely packed things. And people in the group are receiving all sorts of samples and trying things out. And we're also, of course, it's on bioRxiv. Anybody can practice the technique. And we've also been giving a lot of people advice as well. I would love to help people explore this.
Is it sort of I guess a trade-off between what the gel and the introduced reagents will do versus breaking up these kind of dense protein complexes, or is the intracellular environment of the cell always much more packed than the expanded tissue with the extra, chemistry?
Yeah, it's interesting. When we first anchored the proteins to the polymer and expanded the proteins away from each other, I was worried that the polymer would block the antibody from binding because it's anchored somewhere, right, covalently. But to my vast surprise, pretty much all the antibody staining got better. And so, maybe the protein attached to the polymer wiggles around and it's not necessarily constrained in a certain orientation. I mean, I'm being hand wavy here, but it does seem that the benefit of expansion in terms of decrowding more than offsets the additional steric problem of having a thin polymer thread next to it. But yeah, I was quite shocked by that, honestly. So now we're validating expansion and post-expansion staining in a wide variety of other contexts. We're finalizing a manuscript, hopefully any day now. We're expanding human tissues and doing post-expansion staining, and led by Pablo Valdez-Quevedo and others, collaboration with Nino Kyoka's group. So hopefully we'll post that on bioRxiv soon, but we're seeing with decrowding a lot of interesting things in human brain specimens. This hypothesis of decrowding, we're trying to really vet it in a wide variety of contexts.
We have another question in the chat. Using this method, could one determine the adhesion between proteins and/or change in the electrostatic interaction, or determine the change in the interaction force field between the two proteins?
Yeah, great question. So we thought a little about that, but full credit to where credit is due. Bo Wong's group at Stanford published a nice paper in PLOS Biology where they expanded bacteria, and you can use an enzyme called butanolysin to soften it. But if you leave it out, then the bacteria don't fully expand. And they argue that you can use this to make mechanical measurements of what the cell walls of bacteria are doing. And so, it's a really exciting paper. So PLOS Biology, I think they called it mechanical expansion microscopy. So you can do it the regular way and just soften the thing until it expands evenly, or you can leave some of the interactions intact and then use that to make mechanical measurements. So I would expect this idea to be applicable all over the place. Yeah, great question.
Excellent, thank you. And one more from Tom Chang. I assume the expanded difference may be a problem for, say, FRET or fluorescent resonance energy transfer.
Yeah, so we're expanding things away from each other. And at least at the 20 nanometer resolution, we think we're expanding everything away from each other. Again, below 20 nanometers, it's hard to validate, but we're getting there now, and maybe some of these newer hydrogels will get us there. So yes, if something requires close interaction in the final imaging context, that will not work. Yeah, but maybe one could do it cleverly. You could do sort of super resolution FRET, right? Imagine you have some tiny distance and you magnify it, and now it's like FRET had an amplifier on it. I don't know if anybody's tried that yet, but it could be that it could be a problem if one hasn't thought about it strategically, but maybe there might be certain circumstances, and I know very little about this, where a little bit of expansion, maybe not too much, might make the FRET higher resolution. But again, this is not my area of expertise.
I actually also might jump in with a question if that works. So I was reading your preprint that you put out on bioRxiv about whole expansion, where you expanded the whole larva, or the zebrafish larva. And in it, you were doing basically expansion of multiple tissue types in one go, which was very, very interesting. I was wondering how you found the expansion factor between the tissue types post the digestion, and how you validated for that, or if you found there was much difference between like the bone and the other torment tissues.
So if we think of the same paper, this was led by one of our alumni, Jaebum Chang. And so really his group led the way. We helped up only a little tiny bit. So I would have to direct that question to him.
Okay, thank you.
Okay, maybe we could open it up now for questions for any of the speakers today. Does anyone have a final question for anyone who presented today? If you want to put it in the chat, go ahead. Looks like no. So I'm just going to go ahead and share my screen to close things out then. So I wanted to remind everyone tomorrow we'll have another day with 3 sessions. We'll start with a protocols discussion with Brian from Life Canvas Technology. We'll do a session about imaging these types of samples with translucence biosystems and Zeiss. And we will conclude our workshop with a demo data analysis pipeline with Molly and Maria from Arrivis using Vision 4D software. I also wanted to mention that if you have questions or want to speak about anything you saw at the workshop today, you're welcome to join us at our weekly data binge meeting. Nicole will put some info in the chat about this, and you can see some of these same folks at Data Binge and dig into the details for how to get this type of work done in your research project. So with that, I just want to thank everyone for participating today. I thank all the speakers for their time, energy, and expertise. And we will see you again tomorrow morning for our protocols discussion. Thank you, everyone.